field of biology. Today we are honored to have Dr. Kaykhosro Moridi with us. Dr. Kaykhosro Moridi is an expert in the field of medical nanotechnology and molecular biology of the cell and will be sharing their insights and findings on cancer vaccines. We are very much looking forward to hearing their presentation and learning from their vast experience and knowledge. Before we get started, a few housekeeping items. Please ensure your microphones are muted during the presentation to avoid any background noise, and feel free to type your questions in the chat box. We will have a Q&A session after the talk. Once again, thank you for being here. Let's get started and enjoy what promises to be a fantastic seminar.
Hello, hello. Thank you so much for the nice introduction. Can you hear me well? I hope everything is okay. Perfect. So very nice. Now, the thing is, before we even start this session, I'm going to be talking about cancer vaccines. But this is not going to be like a one-sided lecture or talk. I do want you to be participating in here, and I would actually really appreciate it because that would make the whole thing a bit more interactive and fun. Now, obviously, you don't have to. I mean, it's not mandatory to participate, but I would be happy to hear your voices and to hear your opinions. I will be asking some questions, but don't worry, they're not too scientific because, yeah, that's why not we're here. So, anyway, let's start. So, the thing is, everything starts with a problem, and the problem here is cancer. Now, I'm sharing this. By the way, the reason why I'm not like going full screen is that for unknown reasons, when we go full screen, it just stops sharing. So please bear with me. Anyway, so everything starts with a problem, and the problem here is cancer. You guys are all biologists, I assume, and you're already experts in this case. I'm just going to show you some figures. As you can see, you have a lot. This is for 2024, as you can see, and we can easily see that there are a lot of, well, cancer cases, and a lot of them lead to death. This means that cancer is something that should be addressed. This is actually a very big problem. Now, in order to approach anything, we need to understand how it works. Then we would be able to come up with strategies to prevent it, cure it, or fix it, regardless of the situation. You guys know that cancer is when the cells start to grow uncontrollably. This happens because of many reasons, for example, mutations in different genes. Sometimes it's exposure to external stimuli. Sometimes it's just accidents. Regardless of what it is, what happens is that, generally speaking, the checkpoints in your cell cycle go wrong and the cells start dividing uncontrollably. What happens is that then we have cancer. And cancer can actually happen in all kinds of cells, in different tissues. This by itself is a problem because we will actually get to the bottom of this a bit later. But sometimes if cancer is in tissues that cannot be targeted easily, it would cause quite a lot of problems because then you cannot treat it. How would you treat it if you can't target it? Now, the thing is, we do have different means of fighting against cancer. And by that, I mean the tools that we have developed. They're mostly external first. And by that, I mean methods or like tools such as chemotherapy, radiotherapy, or, for example, removal of the tumor. Now, here I'm going to ask you a question. These are, by the way, very effective methods. And as you all know, they're quite popular. They're what we have now. They all share a very unique problem. Now, can anybody either say what the problem is or type in the chat? What do you think all these methods share, the problems that they share? What do you think it is? Any ideas? Is everybody shy, or should I just like... Well, seems like nobody wants to share their ideas. That's fine. The problem with these methods is that they're all invasive. That means that regardless of their good, like say, aspects, what happens is that they also harm the body. For example, in case of surgery, you have a very invasive tool that you're using, and also accuracy is not the best. So there's always that possibility of a recurrence of the cancer. So now we do need to think of a new system, of a new method to deal with it, while trying to reduce the side effects or like negative effects that the other methods have. And that's why we're going to direct our attention to the immune system of our own bodies. Now, generally speaking, our immune system is equipped with great tools to combat anything that is abnormal, like whether it's bacteria, viruses, or cancer cells. And what it does is that it identifies the cancer cells, and it just eliminates them. The problem is that cancer cells, let's say, are somewhat intelligent. What this means is that they come up with the strategies to avoid the immune system. They can suppress the immune system. They can hide from it by just changing their, let's say, membrane surfaces, presenting different antigens, and etc. Actually have a video here, which is quite interesting. I'm actually sure that you cannot hear the audio, the sound, but this is actually a quite interesting video. Let's have a look at it. It's just 1 minute, so bear with me. Now here you have the immune cell, and what it does is actually, well, this is like a T cell, and it's attacking the cancer cells. Now, this is actually a very nice one. It's what happens in your body, and as you can see, what it's doing is that it encompasses the cancerous cells, and very quickly, very effectively, it just eliminates the cell. And the result is that, well, cancer is eliminated, the cell. This is quite good in principle, but the problem is, if it was actually potent, we wouldn't be having cancer or issues related to cancer. Now, that's why we do have this amazing field, which is called immunotherapy. And what immunotherapy does is that it actually uses the body's immune system, its body's own immune system, to fight cancer. The way it does that is that it empowers the body to recognize the abnormal cells. It also boosts the immune cells to fight against cancer. This way, ideally, you wouldn't have any of the side effects that we discussed. This seems like a very good thing to do, a very good thing to happen. So we're just basically, we have a training facility, let's say, hypothetically, for our own body immune cells. And what they do is that they train specific soldiers, potent, to go and fight against cancer. One example is cancer vaccines. Now, we're going to talk about a couple of different things in details, but we're specifically focusing on cancer vaccines and their different parts, the categories and what they do, etc. Now, what happens with cancer vaccines is that, as we talked about it, when cells become cancerous, their composition changes. What it means is that sometimes genes are overexpressed, some genes are eliminated. There are a lot of things that are happening at the, well, let's say, genetic level of the cell. And what this does is that it has an outcome on the surface of the cell. So and the surface of the cell is, the membrane is very important, because that's more or less basically the only way we can recognize cancer cells and tell them apart from our own normal cells. And what this does, what cancer vaccines in principle, generally speaking, do, is that they take parts that are presented on cell surfaces, for example, antigens. We understand how to produce them. Then we expose them. We basically activate B cells and T cells, and then we put them in the body. Everything is presented, by the way, in the picture, in case you cannot follow what I'm saying. But then the activated cells they launch an attack on the tumor cells, and then we will have the tumor death. This is, again, something that can happen, but it's not something which is super potent, and we're going to get into that a bit later. We do have different kinds of cancer vaccines. Generally speaking, there are two kinds of cancer vaccines. They are preventive vaccines, and then we have therapeutic vaccines. If you hear about preventive vaccines, that's usually the rare case, because when we speak about cancer vaccines, this is like in the academic literature, we're usually referring to the therapeutic ones. Doesn't mean that the preventive ones are bad or anything. It's just the case that they're usually more therapeutic. And what they do is that, the preventive ones, such as the ones for HPV or hepatitis, is that they just prevent it from happening. And you probably know it. They're actually very nice and quite interesting. Anyway, what happens with the therapeutic ones is what we just explained. So basically, we just introduce this antigen to the cells, and then to immune cells, and then they take care of the cancerous cells. Now, the question is, first of all, let's understand the differences between normal vaccines, like traditional ones, and cancer vaccines. I'm going to go over a couple of slides all together, and I'll skip the slides when I finish. So just raise your hand if anybody wants to tell me very quickly in brief what normal vaccines are and how they work, if anybody's interested. You can also type. You don't have to open your microphones. Is it because it's like almost 9:30 in Iran, or they make memory? Okay, perfect. Yes. How? Okay, weak or killed microorganisms. Yes, you're both correct. That's very nice. Anything else? Okay, you're both perfectly right. So basically what you said is right. So we have weakened or killed, let's call them agents. They can be viruses, they can be bacteria, they can be like part of them. Doesn't really matter. We introduce them to the body. They are hypothetically going to be, they're going to be unharmful on their own, but they're able to elicit an immune response. And what this does is, as Khadija mentioned, they make the body remember them, so that when the actual pathogen is like introduced to the body, the immune system is able to fight against it and to just get rid of it very quickly. That's brilliant. And we just talked about cancer vaccines. The thing is, now I'm going to go over a couple of slides. The thing is, first of all, with does anybody know the problem with traditional vaccines? Do you know their, let's say, most prominent, most important weakness? How can they be weak? Like, if there was a weakness, what would it be? I'm going to give like 10, 15 seconds for you guys to think about it. They may... Okay, Khadija, do you mean pathogens? Like... Well, the thing is... viruses. Okay, yes, that's actually one of the risks. So we have to make sure that what we're introducing into the body is actually not going to be functional. And that's actually a very specific subject. They're working on it because when you're introducing the viruses, you should be able to introduce it without the harmful part of the body, like the virus, but that's like another subject. But you're absolutely right. The other reason is that they never consider mutations. That's, for example, why we don't have a vaccine for flu, because it just keeps mutating, and we cannot adapt to that. That's a big problem. But also, what happens is in cancer cells, you should consider that cancer cells are quite intelligent because they survive. And by intelligent, I mean they develop means to evade, dodge the immune system. And when that happens, they survive, they harm the body. But if we're considering cancer vaccines, we need to develop something. We're going to get to that a bit later. Whatever we're going to make is still difficult because of the number of different cancer types that we have. And each cancer type has different characteristics. I'm not sure if you're aware of this, but there is almost no specific gene which is mutated in all cancer types. There's always this different set of genes, and they overlap. There are some genes that are mutated in most cancer cells. Then again, they're not specific. They're not common in every cancer. And that's why targeting them becomes a bit difficult, regardless of the method. Now, let's talk about different types of cancer vaccines, and then we will go back to targeting. And I will ask you a couple of questions. We do have different kinds of cancer vaccines. We talked about them. We have the preventive kind, and you can see them here. Like, I'm not going to bore you more than this with them. They're quite well known. Then we have the preventive ones. The therapeutic ones. The therapeutic ones, for example, we have Provenge, which is like for prostate cancer. It's FDA approved, so it's actually functional. It's quite nice. And then we have personalized neoantigen vaccines. Now, first things first. I'm not going to give you the answer. Who knows what neoantigens are? Have you ever heard of them? You can also Google it and just paste the answer. I wouldn't notice. Has anybody heard of them in the past? Going to take that as a no. So what are they? As I said, each cancer has a different set of genes expressed. This means that they have antigens that are specifically presented for them. Oh, okay, they're engineered as pathogens. Well, let's just say that they are antigens which are tumor specific. In simple terms. Now, why are these important? The thing is, you already know it. What happens if I have the most effective drug? Let's say my drug is cost effective, which is, by the way, the most important thing that can happen, like that exists in terms of pharmaceutical sciences. If something is not cost effective, it's never going to happen. It's effective and it has all the good stuff. But if I can't target the cell, it means nothing. Regardless of what I have, I cannot target what I want. And when I can't target my target, I cannot have the effect that I desire. So everything goes south. These are actually very nice because it makes us be able to target anything that we want specifically. Now, how do we work with neoantigens or neoantigen vaccines? What happens is that first we identify a mutation, and these are, by the way, easy work. Now, like in the past, it was quite difficult, but now it's actually quite easy. Then we can produce the neoantigens. We can produce them as peptides, RNAs, or DNAs. And then we will deliver them to the cell. Now they can be dendritic cells, they can be like inside the body, etc. And then what they do is that they basically train the immune systems to attack the cancer through the activation of CD4 and CD8 cells. Now, just a quick thing here. If you has anybody here studied drug delivery, or is anybody here familiar with anything in drug delivery? You can just type a dot in the chat. You don't have to type sentences. Yes. Okay. Khadija, would you be able to tell us how you can target specific cells using different delivery systems? I don't want a specific answer, just a very general answer. By pH. Perfect. So pH is yes. And do you know why we use pH? Let's say in cancer cells. I might be wrong, but I think cancer cell pH is like it goes up to 7.6 inside the cell. Normal cells are 7.2 to 7.4. Yes, but I might be wrong though. You guys feel free to check because but yes, again, the concept is right because they have a different pH. Nice. You guys are right. Overall, the thing with delivery platforms, delivery systems, is that they help us get the therapeutic molecule to the target effectively. And if you study drug delivery systems, you will understand that it's very easy, let's say. I mean, not easy, it's quite possible to modify the surface of whatever you're using so that you can actually go to the target. And it binds, it just enters the cells, it stays in the bloodstream, they can be non-toxic, or even if they're slightly toxic, you can just remove them by adding some particles. We're not going to go deep into those subjects. Anyway, so we can now, the reason why delivery systems are actually very important is because of our next topic, which is RNA vaccines. Everybody is familiar with RNA vaccines, especially after COVID. And what happens is that, as you can see in this picture, we have a specific protein. We design the mRNA related to the protein. We basically produce it. It enters the cells, and then the protein is produced. The antibodies are like go through your immune system. They're presented, and then through the immune system work, you will actually gain immunity to it. Basically, they become activated. Now, the thing is, now let's connect the things that we heard in these two slides. So we have an RNA molecule. Now, okay, so RNA molecules can be a very good solution, but it's not easy to just deliver them to the body. Does anybody know why? What is the weakness of RNA molecules? Let's say they're designed perfectly, by the way, so they will take effect if they're inside the cell. But why do you think that RNAs are not easy to work with? Does anybody know? Perfect. That's because RNA molecules can degrade very easily. They're very sensitive molecules. That's why delivery systems can be very important when we're dealing with instability. Perfect. Exactly. That's the problem. Now, can you tell me how we can fix the instability? Anything? Oh, okay. That's a very nice one. Do you have any ideas? Like, for example, why or how we can use nanotechnology? So we coat, you're saying that we coat the, that's a very nice idea, by the way. But basically what we say, we can say this and then take it. Well, I need to look into that. But you, generally speaking, if you have a molecule, you wouldn't want to change it back to normal again. That's usually a bit time consuming or it's not cost efficient. But I need to look into this. It sounds like a very nice idea. The thing is, very easily we can confine it in like a limited space, then nothing can actually affect it. That's because RNAs are quite reactive molecules. But then if there's no space for anything to react with it or affect it, nothing happens. And that's why, as you guys mentioned, nanotechnology is actually a very interesting, promising field. You have liposomes, you have nanocarriers. Just for your reference, we had CNTs and they were quite popular in this case, but they were ruled off because FDA announced that they're not stable to work with in terms of toxicity. So all the big pharma companies kind of withdrew working with them. They're still like quite interesting, but if you had that idea, I would I would actually recommend you not to think about carbon nanotubes because they're a bit iffy. Anyway, so as we talked about this subject, what happens is that we design the RNA, then we find the right delivery method, we introduce it to the cell. What happens in the cell is that they result in proteins. The proteins are going to in turn activate our immune system. And then, as you guys mentioned, a bit earlier, they will make they actually form memory, which means that if there is a recurrence of cancer, the body quickly reacts, and then we wouldn't have all the bad side effects that can happen. You guys are quite familiar with it. One of the very common cases of relapse is for women and breast cancer. It's quite unpleasant, and it happens quite often. If we can prevent it from happening, that would be like very ideal. So the advantages of RNA vaccines is that we can produce them, we can design them quite quickly, let's say. They're very safe in terms of because of the fact that we're not introducing any kind of pathogens, and if they're released into the body, let's say if the carriers go wrong, nothing is going to happen because RNAs are just going to get degraded. The other thing is that cancer cells can adapt to like anything very quickly. That means that inevitably we will have resistances. But we can easily fix this issue, but by designing a new set of RNAs. And when that happens, we just deal with the cancer. And that's because we can actually design and make these RNAs very quickly. And the immune system that they are able to actually induce is quite strong because they do it through both, as you can see here, antibody mediated way and also T cell mediated one. So you have like a dual effect by which they function, and that's a very nice thing to have. Now, the thing is, we do still have some challenges. We talked about this. The thing is, yes, as you can see, RNAs are quite unstable. There are some side effects. The side effects are actually not that bad because they're like the advantages are just much better. So we can actually, but the side effects are negligible in a sense. But the biggest problem is having a proper delivery system. And what you guys should know is that I'm going to give you like a very quick overview of why making a delivery system is not easy. I'm pretty sure most of you have worked in a cell lab or have seen them. The problem is that when you work in a lab, you're dealing with a couple of cells in a plate. Even if you find something nice, something potent, something useful, that will still be very far from being functional, because the second it is introduced to inside the body, everything changes. There are a lot of other factors that affect it, and that's why a proper delivery system is something that works in humans specifically. Well, I mean, in your target, let's say, but we're talking about humans mostly here. So, for example, a good delivery system should be the reaction. There are reactions. Yes, there will be a lot of different reactions. You're right. So technically, our system needs to go through the body till it reaches the cell, and then it needs to unload its drug load inside the cell, depending on what we're doing. But because we're talking about RNA vaccines, we're just talking about that. But then this whole journey is actually quite difficult and complex, and that's why in a lot of cases, when you reach a solution, like when you have a, let's say, proper delivery system, they just become irrelevant when you introduce it inside the body, and then you will actually have a lot of problems. And then in a lot of cases, you just dismiss them because fixing them wouldn't be cost effective. That's also another problem. So these are the challenges. But on the upside, what we can say overall about RNA vaccines is that they are a very promising area, and there have been great advances in the delivery systems. We have liposomes, we have new generations of actually liposomes. We have different types of them. We have functional nanocarriers. Then we have combination therapies that use different kinds of carriers. There are also every day a new carrier is emerging, which makes RNA vaccines something worth working on when dealing with cancer. So basically, if you guys want to think about this subject, I would say that designing and coming up with a potent carrier is relatively more important at the moment than designing the mRNA itself, because that's quite easy. You just need to identify the mutation that you want. Sometimes it can be challenging, but generally speaking, that's easier than designing the carrier, which is functional. We do have different other kinds of vaccines. One of them, for example, is oncolytic virus vaccines. And they're actually quite interesting because what they do is that they have they act on two different fronts, let's say. These are either natural or engineered viruses, and they're very selective. They infect and kill cancer cells, and they're just indifferent to normal cells. What they do is that first, I mean, technically simultaneously, they replicate within the cell, and they do it to the extent that the cells burst, and then they also release antigens which stimulate the body's immune system. Then so you have the other that you have the immune system fighting against the cells while the virus is actually killing the cells from inside. And you also have viral vector vaccines. Now, generally speaking, you can refer to them as carriers, because these are just modified viral vehicles, and they just deliver the genetic material to the cancerous cell. And that's basically it. That's how they work. They're quite nice, but the problem is that they're not super stable, and designing them sometimes has proven to be a little challenging. Nevertheless, they're still quite interesting to work on. Now, with this slide, I would like to conclude my talk. I wanted to actually say that this was a brief introductory talk about cancer vaccines. This is a very, very big subject. There is a lot of research going on, and there's a lot of information that I excluded. I am going to actually talk specifically about COVID and COVID, the vaccine that we have. There's a lot of controversy behind it. There's a lot of things that some people don't know. And also, it's always good to know the mechanisms, because that's a very, very fast growing part of the cancer vaccines, and they're actually becoming very popular these days. Now, I'm going to ask you to participate in this section. And based on the things that we talked about today, what do you think? How do you think we can overcome all the challenges that we have with cancer vaccines? Maybe you guys have read some new articles. Maybe you've heard something from someone. Maybe you've watched videos. Does anybody have any ideas of how we can overcome, let's say, some of the challenges that we have in case of cancer vaccines? Feel free to speak, by the way. You can also speak in Farsi. You don't have to speak in English. That's perfectly fine. I would appreciate your cooperation. Just going to give it like 20 seconds to see if there are signs of life from anyone. Okay. Okay. Can you explain that a bit more? That's a very nice one. How can we do that? This is going to be a very long, awkward silence because I'm just looking at the chat and just waiting for a response, by the way. While you're typing, I'm going to give everyone an overview of how we can overcome the challenges. So first things first, one thing that you need to be aware of is that doing research in this case doesn't necessarily have to be leading to success, because in a lot of cases, by eliminating things that wouldn't work, we're actually facilitating everyone's work to reach something useful, to find a useful solution. We can use tools like CRISPR-Cas9. That's actually what? Okay, that's a very yes, exactly. CRISPR-Cas9 is actually a very, very nice one. Well, for those who are not familiar with this, think of it as like a pair of scissors. We just cut whatever we want in the genome. That's basically it. It's very difficult though. Like, it's not as easy as I'm explaining it. But the CRISPR-Cas9 is actually, and if you're considering that, if you're considering to work on that, there are hundreds of positions for it, and it's a very hot topic these days. And it's by the way, it's very functional. The only problem with CRISPR-Cas9 is like the accuracy that it has, but that will be fixed. Like, the resolution will be fixed in no time. So you're absolutely right. We can use CRISPR-Cas9. We can just genetically just remove the genes. The problem is, like, basically, like, very precisely, the problem is that you can't really be doing that when the cancer has already, like, it's like in later stages. So it is very effective, but you need to use it in the right place at the right time. Nevertheless, it's just something quite interesting. Generally, you guys need to consider a couple of things when working with, let's say, RNA vaccines. First, they should be delivered effectively. Then they should actually be able to deliver the function that you desire. They need to be, the whole system should be cost effective. That's basically it. So I would encourage you to actually take a look into finding different carriers and how they work. They're actually a very, very nice field. We actually have like a lot of articles on different carriers for RNAs. And that's it. If you have any questions, I think we do have a section for Q&A. If you have any questions, if I know the answers, I will be more than happy to help. So I thank you, everyone, and I wish you that small Ferrari, the bottom right of my screen. Thank you so much for the participation. If you have any questions, I'm at your service. I'm just going to wait for a couple of minutes, maybe, to see if anybody has a question. And thank you, everyone, for participating, because I know it's quite late in Iran, and nobody wants to study this late or hear anything scientific. My email. Sure. This is my normal email. If you have any questions, you can ask me. Okay. Well, thank you so much, and I hope you have I'm glad you liked it. I hope it was useful, and I hope everybody has a great night, and I hope to see you later in my other talks. Have a great night, everyone. Bye-bye.